Now we're going to look at lipids, another type of large biological molecule. But remember that unlike the other large biological molecules, lipids do not form polymers. So they are not made of a monomer, repeatable subunit building block material. In fact, lipids are pretty diverse, but what they all have in common is that they are hydrophobic molecules. Hydrophobic means that they have little or no affinity to water, or they repel water, or are repelled by water. It has to do with the structure. Mostly they are hydrocarbons that form non-covalent bonds. So that also makes them nonpolar. So they don't want to interact with water because they're already fulfilled with their valence of electrons. This group, again, of very diverse molecules, contains several different types of lipids that are each biologically important in their own way. And these are the fats, the phospholipids, and the steroids. So this is how we're going to be studying the lipids, is by looking at each of these groups. And we're going to start with the fats. Fats are a molecule of energy storage for a living thing. Kind of similar to carbohydrates, except the structure is much different. You don't have monosaccharides here. Instead, you can see what kind of stands out to me when I see this diagram at the bottom would be the fatty acid tail, that long chain of hydrocarbons, and that is one component of a fat. So fats let's start with an understanding of the structure of a fat. Fat has a glycerol molecule, which is the one on the left in the gray box, that's attached to 3 fatty acids. The fatty acids are the long hydrocarbons, and notice that the point of attachment where you have a hydroxyl group on the fatty acid and a hydroxyl group on the glycerol, through dehydration synthesis is how these can become connected through a covalent bond. So again, we're seeing the role of dehydration synthesis in the formation of these molecules. Even though this one isn't a polymer, that process is still needed to synthesize these molecules. Now this one isn't shown in full completeness. There could be the 3 fatty acids attached to that glycerol, and that's what would make it a fat. And we'll see that on the next page because there's different types of fats based on the structure of the fatty acids. Fatty acids can vary in length because of the different numbers of carbons and also the number and locations of double bonds. Perhaps you've heard of fatty acids that are saturated versus unsaturated. Well, what does that mean? Chemically speaking, a saturated fatty acid has the maximum number of hydrogen atoms possible. So when you're looking at the fatty acid tails here, which are just shown as these zigzag lines, each point, each intersection would represent a carbon. And the way that these are depicted would indicate that the carbons are fully saturated with hydrogens because there's no double bonds. They would just have their valences completed by sharing electrons with hydrogens. It's just a simplified way of drawing chemical structures. Maybe you'll learn about that more if you take an organic chemistry class. So in contrast to the saturated fatty acid where you see those zigzag fatty acid tails, single bonds, notice that in the unsaturated fatty acid, there is a double bond here, shown where my arrow is pointing. So here you would have carbon, carbon, and there would not be full saturation with hydrogen because they're sharing some of their electrons with the carbons. Actually, 2 pairs of electrons would be shared with the carbons. So anytime you have 1 or more double bonds, then that would now deem it as an unsaturated fatty acid. Okay, so that's the definition of what makes it a saturated versus an unsaturated fatty acid. But how do these differ in their roles or how they can affect a living organism differently? This is a very important thing to know because it can affect your health. Saturated fats are fats that are made from saturated fatty acids. A way to tell if something has saturated fats is to leave it at room temperature, like a stick of butter, and you're not sure if that butter is made from animal fats or plant fats. So if you leave it at room temperature and it stays solid, it doesn't melt, then it does contain saturated fats. Some studies have shown that a diet that's rich in saturated fats may also contribute to heart disease through plaque deposits forming inside of our arteries and blood vessels. So if you're found to, in blood work, potentially have like high cholesterol, that would indicate that you, or blood pressure, that could indicate that you're maybe getting a little too much of the saturated fats because it's forming that plaque. And the reason it's doing that, if you look back at the chemical structure, those saturated fats can pack together pretty tightly. Since there's just those single bonds holding those fatty acid tails together, they can lay nice and compactly inside the lining of our bodies where those molecules pass through. They start to kind of just get stuck and then stick there more and more. Versus the unsaturated fatty acids, with that little kink in the tail, there's a little bit more wiggle room for those molecules. And so they don't tend to lead to the same health problems that consumption of a high amount of saturated fats would. The unsaturated fats that come from plant fats and fish fats would be liquid at room temperature. And we also would call this group oils because of the fact that they are liquid at room temperature. So some studies again have shown that it's better to consume more of these unsaturated fats than saturated fats. Let's go on to another type of lipid, the phospholipids. They're similar to fats, but instead of there being 3 fatty acids attached to a glycerol, we're going to see there's only 2 fatty acids. Like before, the fatty acid tails are hydrophobic, again, because they're mostly composed of hydrocarbons. But in addition, there's going to be a phosphate group that gives a property to this molecule of having a hydrophilic region and a hydrophobic region, because the phosphate it's an ion, it's PO4 -3. So this molecule is different in that respect, has polarity. So why is this significant? If you had phospholipids in an aqueous environment, they're going to self-assemble into a bilayer where the hydrophobic tails are all pointing towards the interior, trying to get away from the water that's around them. And all the hydrophilic regions are going to be on the outside, near the water. So this is a unique thing that happens without any living thing necessary, no energy required. It's just because of the chemical nature of that molecule. But to bring this back to biology, phospholipids are the major component of all cell membranes. So let's take a quick look at that, just so you have an idea of where we're going to be going with this in a later chapter. So again, the nature of these molecules is that they have a hydrophilic region and a hydrophobic region. The hydrophilic region would be facing the water because it can interact with water, it wants to interact with water, but the hydrophobic tails are all huddled together, trying to get away from that water environment. So that's why we see the bilayer here. All the tails from the 2 layers are facing inward at each other. And by the way, these are only held together by the weak hydrophobic interactions of those tails. There's no hydrogen bonding, covalent bonding, ionic bonding here. So it's actually pretty easy for this to be ruptured, or even for molecules to kind of get in and around those phospholipids to be able to pass through a cell. Or we'll talk more about that in a chapter about the cell when we learn about the organelles, and then get into the membrane itself. Okay, we have one more type of lipid group, and those are the steroids. So steroids, different in structure from the two groups we've just studied, fats and phospholipids. Um, Carbon skeleton composed of 4 fused rings is what defines these steroid molecules. Here are some examples of steroids. Cholesterol, which is a component in animal cell membranes. Again, we'll learn about that when we study the membrane in chapter 5. And hormones, like testosterone, the primary sex hormone in males. These are also a type of lipid. So although these aren't involved in energy directly, it is important that these steroids are present in animals, especially because they're involved in maybe cell-to-cell communication. And they have other roles, like in hormones. It could be for sex, for development purposes, for communicating within or even outside of a cell or a living organism. So there's many different, it's a very diverse group. We're just looking at a few specific examples. And some of these, again, we'll circle back to when we study in more detail the cell membrane in particular. Okay, but that concludes the lesson today for the lipids.